Okay, so get this. Scientists may have found a way to, like, actually control how our cells age.
Wow.
Yeah.
That's pretty amazing.
It is, and we're going to deep dive into this research
Yeah.
from Osaka University.
Yeah.
You're not going to want to miss this. So they found a specific protein that appears to be really important in controlling aging at, like, the cellular level.
Right, and so much of aging is about what's happening in our cells.
Totally.
So this is really exciting stuff.
It is.
Yeah, so it really kind of centers around these things called senescent cells, which are basically cells that have gotten a bit, shall we say, tired? They've slowed down. They've stopped dividing like they used to. And they just tend to build up as we age.
So it's like the cellular equivalent of hitting that snooze button one too many times.
Exactly.
Yeah.
But here's the thing. These senescent cells aren't just taking it easy.
Wow.
They're also quite literally bulking up.
Yeah.
They're physically larger than their younger counterparts. And their internal structure is different, especially in these areas called stress fibers.
Okay, hold on, stress fibers.
Yeah.
Can we break that down a bit?
Sure.
Sounds like something you'd find at a microscopic gym.
You're not far off.
Oh.
So imagine the inside of a cell, like a bustling city, with all sorts of structures and pathways.
Okay.
Stress fibers are kind of like the scaffolding that helps maintain the cell's shape and allows it to move and interact with its surroundings.
I see.
So you can think of them as kind of internal support beams, but they can also be involved in, like, communication and signaling within the cell.
Gotcha. So in these older senescent cells,
Yeah.
these support beams are getting, like, beefier.
Precisely.
Okay.
And it's a bit of a mystery.
Ah.
Even the researchers were puzzled by how these cells managed to maintain their larger size.
Oh.
The lead author of the study, Pirawan Chantachodakul, even said, "We still don't understand how these senescent cells can maintain their huge size."
Wow.
Yeah.
Okay, so how'd they figure out what was causing this cellular bulk up? What's the protein of the hour?
Enter AP2A1.
AP2A1.
Yes, this protein is found in much higher amounts within those stress fibers of senescent cells.
Oh.
The researchers had a hunch that it might be the key, so they decided to do some experiments.
Okay.
They took old cells and removed AP2A1.
Oh, wow.
And then just to see what would happen, they added it to young cells.
Okay, so they were basically playing cellular puppet masters
Exactly.
with this AP2A1.
Yeah.
What happened when they pulled the strings?
Well, the results were, to put it mildly, astounding.
Okay.
When they suppressed AP2A1 in those older cells,
Uh-huh.
some signs of aging actually started to reverse.
Oh, my God.
The cells were behaving more like their younger selves again.
Wait, are you telling me they essentially hit rewind on those cells just by messing with this one protein?
It really does seem that way.
Wow.
And to confirm their suspicions, they took those young, healthy cells and flooded them with extra AP2A1.
Okay.
And guess what?
I'm on the edge of my seat here. They sped up the aging process.
You got it.
Oh my gosh.
The young cells started exhibiting characteristics of premature aging.
Wow.
It was like they aged decades in a petri dish.
Wow.
So it's pretty compelling evidence
Yeah.
that this AP2A1 protein is a major player in regulating how our cells age.
So this isn't just some random correlation. They're directly manipulating the levels of this protein and seeing clear changes in how the cells age.
Exactly.
That's huge.
Yeah.
But how exactly is this AP2A1 working its magic at the molecular level?
Right.
What's the mechanism behind all of this?
That's where things get even more fascinating.
Okay.
AP2A1 doesn't work alone.
Oh.
It has a partner in crime,
Uh-oh.
another protein called integrin 1.
Integrin B1.
Yes.
All right, taking notes here. Tell me more about this protein duo.
Okay.
What are they up to inside the cell?
Well, think of integrin 1 as the ultimate cellular glue.
Okay.
It helps cells attach to the collagen matrix that surrounds them, which acts like scaffolding to give tissues their structure.
So AP2A1 and this integrin bear 1 are working together like a construction crew to make sure the cell is properly anchored to its surroundings.
That's a great analogy.
Yeah.
And remember those stress fibers we talked about earlier?
Yes.
Both AP2A1 and integrin VER1 are found along those stress fibers.
Oh.
Researchers believe that as these proteins move along the stress fibers, they might be strengthening the adhesion,
Okay.
the grip that the cell has on the surrounding matrix.
So more AP2A1 means more of this integrin bear 1 glue.
Right.
Which means a stronger grip on the matrix.
Yeah.
And that's why the senescent cells are getting bigger.
That's the current thinking.
Why?
Like the cell is saying, I'm not going anywhere.
Oh, my God.
And that stronger adhesion,
Yeah.
that stronger connection to the matrix, could be contributing to the increased size of those senescent cells.
It's like they're clinging on for dear life and spreading themselves out.
That's a great way to visualize it.
Yeah.
And it's not just about size. These changes in adhesion can also affect how the cell functions, how it signals, how it responds to its environment.
So AP2A1 is influencing integrin burla, which is impacting focal adhesions, which is ultimately changing how the cell behaves and ages.
Right.
It's like a chain reaction.
It is.
With AP2A1 as the initial trigger.
Exactly.
That's pretty amazing.
Yeah, and the more we understand about this chain reaction, the closer we get to potentially intervening in the aging process itself.
Wow, I'm already starting to see the potential implications here. If we can control this protein AP2A1, could we potentially control the aging process itself?
That's the million-dollar question, isn't it?
Wow.
And it's not just science fiction anymore.
Wow.
This research is opening doors to possibilities we could only dream of before.
This is definitely one of those deep dives that makes you realize just how quickly science is moving.
Yeah.
We're not just talking about slowing down aging. We're talking about potentially reversing it at the cellular level. What are some of the other ways this discovery could be applied?
Well, for one, imagine being able to use AP2A1 as a kind of cellular age meter.
Oh.
Instead of just relying on someone's birth date, we could analyze their cells and see how much of this protein they have to get a more accurate picture of their biological age.
So we're talking about moving beyond just chronological age.
Yes.
And actually understanding how old our cells are on a deeper level.
Right.
That's incredible.
It is, and it gets even more exciting when we start to think about therapeutic applications.
Okay.
If we can target AP2A1, maybe we could develop treatments to slow down or even reverse the progression of age-related diseases.
Wow.
Imagine the possibilities.
All right, now you're talking about some truly mind-blowing stuff.
Yeah.
We're talking about potentially extending human lifespans and improving quality of life in ways we've never imagined.
It's pretty remarkable.
But with such a powerful discovery comes some serious questions, right?
Absolutely.
What happens to our concept of mortality? If we can significantly extend human lifespans, how do we ensure equitable access to these treatments?
Those are all incredibly important questions, and they're not easy to answer.
This is definitely a deep dive that's going to stay with me for a while.
I think so.
But before we get too lost in the philosophical implications, let's bring it back to the science.
Okay.
You mentioned this integrin E1 protein that's working alongside AP2A1.
Yes.
Can we dig a little deeper into the nitty-gritty of how these two proteins are interacting?
We can certainly do that.
Awesome.
Integrin Weta is a fascinating protein in its own right,
Okay.
with a complex structure and a whole host of functions within the cell. So it's a transmembrane protein, meaning it spans the entire cell membrane,
Okay.
acting as a bridge between the cell's interior and the external environment.
So it's like a doorway that lets the cell interact with the world outside.
Exactly. And integrin E1 isn't just sitting there passively. It's actively binding to molecules in the extracellular matrix,
Okay.
particularly collagen. And this binding triggers a cascade of signals within the cell, influencing everything from cell shape and movement to gene expression and even cell survival.
Wow, so it's not just about physical adhesion.
Right.
This integrin bulmun is like a communication hub,
Yeah.
relaying messages between the cell and its surroundings.
You got it. And this is where things get even more intriguing.
Okay.
When integrin bulboni binds to collagen, it actually clusters together with other integrin molecules, forming what are called focal adhesions.
Focal adhesions sounds important.
They are.
What are those exactly?
Think of focal adhesions as anchoring points where the cell firmly attaches to the matrix.
Okay.
They're like the cell's strong grip, allowing it to resist forces and maintain its position.
So it's like the cell is using these focal adhesions to like really dig in and hold on tight.
Exactly. And these focal adhesions are incredibly dynamic.
Okay.
They can assemble and disassemble, grow and shrink depending on the needs of the cell.
Wow. So it's not just a static connection. It's a constantly changing responsive system.
Precisely. And this dynamism is crucial for all sorts of cellular processes, from wound healing to immune responses.
Okay, so we've got this integrin MEL1 acting like a cellular glue.
Yes.
Forming these dynamic focal adhesions and relaying signals between the cell and its environment.
Right.
But how does this all tie back to AP2A1, and the aging process.
Well, here's where the pieces of the puzzle start to fit together.
Okay.
Remember, AP2A1 is found in higher amounts within the stress fibers of those senescent cells.
Yeah.
And those stress fibers are connected to those focal adhesions, those anchoring points we were just talking about.
So AP2A1 is somehow influencing how integrin male 1 is working.
Yeah.
How those focal adhesions are forming and functioning.
That's the hypothesis, it seems, that the increased levels of AP2A1 might be leading to stronger, more stable focal adhesions.
Okay.
And as we discussed earlier, that stronger adhesion, that stronger connection to the matrix, could be contributing to the larger size of those senescent cells.
It's like they're holding on for dear life. And that grip is making them spread out and bulk up.
That's a great way to visualize it.
Yeah.
And it's not just about size. These changes in adhesion can also affect how the cell functions, how it signals, how it responds to its environment.
So AP2A1 is influencing integrin mate 1, which is impacting focal adhesions, which is ultimately changing how the cell behaves and ages.
Exactly.
It's like a chain reaction with AP2A1 as the initial trigger.
Exactly. And the more we understand about this chain reaction, the closer we get to potentially intervening in the aging process itself.
That's incredible. We're not just observing aging. We're starting to understand the molecular mechanisms behind it.
Right.
And that opens up so many possibilities. So before we get too far ahead of ourselves, let's zoom out a bit.
Okay.
We've been talking about this research in the context of aging, but are there any other areas where these findings might be relevant?
That's a great question. Remember, integrin maple is involved in a wide range of cellular processes, from cell growth and migration to immune responses and tissue repair. So anything that affects integrin function could have implications beyond aging.
So we're talking about potential applications in wound healing, cancer research, immunology.
Exactly.
List goes on.
The list goes on. And that's what makes this discovery so exciting.
Yeah.
It's not just about aging. It's about understanding the fundamental processes that govern how our cells behave and interact with their environment.
It's like we've stumbled upon a key that unlocks a whole new level of understanding about cellular biology.
I like that.
And that understanding could lead to breakthroughs in so many different fields.
I agree.
But I have to ask, with such a powerful discovery, there must be some challenges ahead, right? It can't all be smooth sailing from here. What are some of the obstacles researchers might face as they try to translate these findings into real-world applications?
You're right, there are always challenges when it comes to translating basic research into practical solutions. In this case, one of the biggest hurdles will be developing therapies that specifically target AP2A1 without causing unwanted side effects.
Right, because we're talking about a protein that's involved in so many different cellular processes.
Yes.
Tweaking it in one way could have unintended consequences elsewhere in the body.
Exactly. It's a delicate balancing act.
Yeah.
Researchers will need to carefully study how AP2A1 interacts with other proteins and pathways to ensure that any interventions are both safe and effective.
So it's not just about finding a drug that targets AP2A1. It's about understanding the entire network of interactions and finding a way to modulate it precisely.
Precisely. It's a complex challenge, but the potential rewards are immense.
Yeah.
Imagine being able to develop treatments that not only slow down aging, but also address a wide range of age-related diseases. From heart disease and cancer to Alzheimer's and Parkinson's.
We're talking about a paradigm shift in healthcare, a move away from treating individual diseases and towards addressing the underlying process of aging itself.
That's right.
But even if we manage to overcome these scientific hurdles, there are still some pretty big ethical and societal questions to consider, right?
Absolutely.
What happens to our concept of mortality if we can significantly extend human lifespans?
Right.
How do we ensure equitable access to these treatments?
Those are incredibly important questions, and they're not easy to answer.
This research forces us to confront some fundamental issues about what it means to be human. About our relationship with time and mortality, and about the kind of future we want to create.
It's a lot to think about.
It's a lot to wrap our heads around.
Yeah.
But that's what makes this deep dive so fascinating.
Right.
It's not just about the science. It's about the implications of that science for our lives, our societies, and our future.
Absolutely. And while we don't have all the answers, one thing is clear.
What's that?
This research has the potential to transform our world in profound ways.
It's truly mind-blowing. We've journeyed from the inner workings of cells to the potential future of humanity. And it all started with this one protein, AP2A1. I'm still trying to process everything we've covered. Do you have any final thoughts you'd like to share before we wrap up this incredible deep dive?
It's a balancing act, isn't it? Embracing the incredible possibilities of science while also being mindful of the potential consequences.
It's like we're standing on the edge of a new frontier, and we need to proceed with both excitement and caution.
I think that's a great way to put it. This research into AP2A1 and cellular aging is just one example of how science is forcing us to rethink our assumptions about the world and our place in it.
It's almost like science fiction is becoming reality right before our eyes.
It really is.
And speaking of science fiction, it's hard not to think about all the stories we've read and watched about the implications of extending human lifespans. What are some of the societal shifts we might see if people start living significantly longer?
Well, imagine the impact on workforce demographics, retirement planning, healthcare systems. It's a complete paradigm shift. And then there are the deeper philosophical questions. How does our understanding of life and death change if we can significantly delay the aging process?
It's mind-boggling, and it's not just about individuals. It's about how societies as a whole adapt to this new reality. Will we see new social structures emerge? New forms of art and literature, new ways of thinking about time and purpose?
These are all questions that we as a society need to start grappling with. Now it's not enough to simply let the science advance on its own. We need to have these conversations about the potential consequences, both positive and negative.
And it's not just about predicting the future, it's about actively shaping it. We have a responsibility to ensure that these scientific advances benefit all of humanity, not just a select few.
I completely agree. We need to be mindful of issues like access to treatment affordability and potential disparities. We don't want to create a world where only the wealthy can afford to extend their lifespans.
It's a reminder that scientific progress isn't just about technological advancement, it's also about social justice and ethical considerations.
Exactly. And it's not just up to scientists and policymakers to figure this out. It's a conversation that needs to involve everyone. We all have a stake in shaping the future, and we need to be informed and engaged citizens to ensure that these powerful discoveries are used for the greater good.
So for our listeners out there who are now pondering the future of humanity along with us, what's one final thought-provoking question they should consider as they continue to explore this topic?
I'd ask them to think about this. If we could choose to slow down or even reverse our own aging process, would we want to? And if so, at what cost? It's a personal question, but it also has profound societal implications. Our individual choices will shape the future. So it's crucial that we approach these questions with thoughtfulness and a sense of responsibility.
Wow, that's a powerful question to leave everyone with. It's been an incredible deep dive into the world of cellular aging, the potential for reversing it, and the mind-blowing implications for the future. Thanks for joining us on this journey of discovery, and until next time, keep those brains buzzing.